Hi, Clementine.
Hi, how are you?
I hope others will join too, but I noted there was no um, agenda made, so I made a very short one this morning.
Yes, yeah, I noticed. I think because of that the one last month being cancelled, I think everyone's gotten confused. Um
Yeah, and it feels
you-
like this one is earlier as well, because it seems it's not the last Tuesday, is it?
no, no, you're right. That hmm,
Yeah,
I
next
wonder
week is the
why.
last Tuesday.
Yeah, we've got the 29th.
Yeah, but
Well,
somehow it ended up like this in the agenda, so I thought let's stick to the agenda.
yeah, I mean, doesn't make much difference to me.
So maybe it's the fourth Tuesday, something like that.
Yeah, that maybe that would explain it. Because let's see. Yeah, it is the fourth Tuesday. And I suppose last time we had it on the 25th. Yeah. It must be the fourth, not the last.
Yeah, but in general, it's the same. But yeah,
Yeah. Hi, Matt.
Hello. How's everybody doing?
busy with the rebuttal.
Yeah, absolutely. It's a big one.
Yeah, and it's
Well,
also a lot of text, um,
yeah,
which was added by others and which I
yeah.
tried to make as concise as possible. But
I know
Is
it's
that
a hard one.
is that Mickey's rebuttal or what?
yeah,
Yeah.
the kinase and the due date is tomorrow.
Oh no. Okay.
Yeah, I mean,
So
I think the modeling bit does definitely help because an attempt's being made to figure out what's going on. I think it just needs to be slightly, you know, uh, maybe just described differently. So slightly better connection between what's observed and whether the modeling supports that. And then it definitely does add something and definitely does help to respond to the referees, I think.
yeah, the only part is that I don't get much response from the modeler, so that usually
Ah,
delays
right.
things.
Okay.
So
Yeah.
yeah,
Well,
maybe I need to submit it like it is. I did do the textual things and then go for the second rebuttal to make this conciser.
mm, mm. I can I can give an update on the Phenomenon paper because Clementine and I were just corresponding about it. Um, if we want to just dive in, I mean, if
Yeah, I was gonna say I'm happy to take minutes. And then if people, you know, join later or have missed it, they can catch up
cool,
later then.
cool. Okay,
Yeah,
great. So there were just what was it? There was the one thing that I was just dealing with is the last couple of things. I mean, it's always the way, right? The last couple of things to do with compounds that are in the paper that we didn't quite have all the data for. And that was one of Kimberly's compounds from Sydney way back. And I realized why we didn't have that. It's some neutral form of a salt that we use quite a lot in the paper. And normally you make this salt form, it's the salt of an amine, and then you use that in subsequent reactions. And the reason you do that is because keeping an amine as a salt form can keep it nice. But we did actually generate the neutral amine, which has a tendency to get oxidized and go off as with any other amine. And we did test it. And so because we tested it and because it's in all the diagrams, we should have some characterization for it. So I found that actually that is what this compound is. Kimberly's compound is this neutral form of an amine, which we already report. So um, we thank you very much, Clementine, for all your detective work. We found the relevant procedures, and there was some NMR data. And Clementine and my student have been looking at the NMR data, and we figured out that it looks okay, and so we can include it now, and we're all done. So I've just inserted that last compound into the SI, which is great. It was a very minor thing, but we had to resolve it one way or the other. And if we didn't include it, we'd have to take it out, right? And then the other thing which I've been waiting on is the compounds from IMSA that we had contributed. And there were 9 originally, but many of those actually came from another source as well. So we can include the data through somebody else having made the compounds. But there were 3 compounds that were just made by IMSA. And, this is one of these organizations that's contributed stuff. It isn't a big main MycetOS partner. And with any open project, right, you've always got this issue that people might contribute something and then maybe drift away or whatever. And so we didn't have data for those compounds. So I tracked down the LCMS data, which shows the compounds are fine. And then I asked John, the guy who led that team, for the NMR data, and he sent me the listing for the compounds. So the sort of the letters and the numbers for it, but not the actual NMR data, which is what we have for everything else, and which is kind of the typically the level of detail typically required. So what I'm going to do is include them in a little section at the end of the SI, and I've just got to paste in the data into a separate section saying, these compounds were contributed by this team, and just to sort of make it slightly separate. And I think that'll be fine. If anybody complains about it, we can delete them. But the LCMS shows they're pretty good, and I think it's okay.
Okay.
So that's it. And then I've just got to. It took me like an hour just to reorder all the NMRs in the SI. So I was doing that, and then I've just got to sort out the formatting of all of that because it's all a bit haywire, and then put some references in, and then I'm done, and then I can submit it. So that is genuinely this time. That is it. I know I said that last time, but that is now it. There's just lots and lots of fiddly things. And it's not a big surprise because there's a lot of molecules in this paper and they've been contributed by multiple people. So there's always complexity. So I will hopefully be able to get that submitted today if no crises happen.
Okay.
Which would be great, right? Because we're talking, in this meeting, I guess we're talking about these 3 papers, right? And if we get them submitted, that's 3 more MycetOS papers, and that would be a big deal for us, I think.
Yeah.
Okay, that was my update.
Yeah, so the kinase rebuttal I just mentioned, I'm working on it, and it needs to be submitted today to beat the deadline. So I guess it needs a little bit refinement later on in the accepted stage or in a second rebuttal, simply on the modeling. Also, I will need to get better figures from the team in India upon when it's accepted on publication, because the current supplementary figure 7 and main figure 6 are a little bit too blurry. But simply I don't have the better figures at this time, so we will need to supply them when it's accepted. And the benzimidazole paper, I saw that many of the chemists, so Andrea worked on the chemistry part of the manuscript. I simply need to go through to see what else is changed, to see if we can make a final version of that. But it seems also nearly there.
Yeah. So just on that, I had a couple of hours to spend on a flight on holiday, and I thought, right, I'm offline. I'm just going to look at this data and try and make sense of it. So I made this figure, which, if you just hang on a second, I might just share it if that's okay, just because I need to ask a question about this. Hang on. Okay, hang on. Okay. Can you see the molecules?
Yes.
Okay. So this is all of them. So the ones that are highlighted in the paper are up top to show those structures, but they are the green ones up top here are those active ones. Okay, so they're included again. And the reason I wanted to make this diagram is to try and show well, to try and see if there was anything in that we could say about the structure-activity relationship for these compounds, because I couldn't really tell what that might be just from the tables. And I think and these are arranged, I haven't actually done the final rearrangement. So the little tiny numbers in gray are, I did the sort of usual traffic light thing with the green being better and orange okay and red bad. And then there wasn't quite enough detail. So I've included also the kind of average, I think it is, percent growth at 25 micromolar, I think it is. The kind of most interesting column for which we have data for all compounds. And I put those in there, and that might lead to a slight rearrangement of things, but as it stands, it's pretty good. And the thing that is, well, there are two things I think that stand out. One is that this, I think, pretty well shows that aromatic substituents are better, right, than allocyclic. So all of the ones in orange up in the middle of the diagram are the aromatic rings, and all the ones in the bottom, which are lower potency, are the other things like not aromatic rings. I mean, it's pretty clear to me. And there is quite a lot of tolerance of change. I mean, lots of different molecules here are getting pretty good values, I think. But the other thing is, so that's one conclusion that I think I'll put in an email.
Yeah, is this the Madurella mycetomatis data, or is this both?
Yeah,
because yeah,
I could look at the others, I suppose, and check it's the same, but I'm not sure. Yeah, I mean, this is just the mycetomatis data. Yeah.
Yeah. Okay. Because there are differences between the two.
Right. I might do a spot check of that then and see if that changes the message. I'd be surprised, but I can check that. But the other thing is, if you look across, so on the left is the chloro substituted, on the right is the bromo substituted, right? And if you look across at each row there, and I just arranged it by similarity. There's no particular difference between rows 3 and 4. It's just I've arranged it so that the potencies roughly track. You can see that there are, I mean, there's a great deal of similarity between the two. So having chloro and bromo in blue up top doesn't really make any difference, right? And that's quite useful as an internal check, because the chloro is one and the bromo is another, and they vary by that atom, but it doesn't make any difference. But the values that you're seeing for both compounds track quite well, which is good for reproducibility. I think it's still worth keeping the chloro panel and the bromo panel. You could make an argument for just merging the two somehow and saying, well, you know, because there are lots of common structures, you could make an argument for that, but I think it's okay leaving it as it is. I mean, you can see the duplication sometimes, but I still think it's kind of useful to have all the numbers there laid out like this, and it does act as a sort of internal check that these numbers are believable. But I can raise that in the email too, because I think that the other two may have some, you know, suggestions about that. The one that's the standout though is 527 here with the chloro here and this cage thing called an adamantane, which is one of the highlighted compounds. It was one of the ones that did really well. You change to the bromo and it's way down here, right? Completely inactive. There's nothing else like that in this data set where you go from very potent to completely inactive. And you can see how well they track. So I suspect that one of those, well, those two compounds ideally should be retested. I know we can't do that necessarily, but that's like a red flag.
Matt, these are in vitro assays. So the less than 30% is actually not the criteria we use. So you could make it less than 20%, but not progressed. Because I think the majority of the compounds, except one, are in that category less than 20%. So that would normally be that they would proceed to in vitro, to in vivo, or further in vitro characterization. The reasons why these orange ones were not progressed was because they most likely were not active against falciformis spora senegalensis. So that doesn't mean that they are not active against Madurella mycetomatis.
Sure.
It simply means they were not selected because they were not active against the other fungal species. But that makes your stoplight maybe a little bit more easier and your conclusion a little bit stronger even, because most of the orange will be green. Because looking at the values, the 16 is the percentage growth, so the gray ones. So there's only 1 or 2 orange ones on the left side, which then would be red. And that's also like, for instance, the one with OME on the top, which is red with the brome one, and orange with the chlorine one. They would be both red then.
Yeah, right. Okay. But yeah, I could definitely change that.
That's the biological criteria we usually use. Yeah.
Right. Yeah, and the progression thing, I mean, that was just my looking at the sheet and seeing which compounds had been tested further. So the progression there was yeah, just because there were fewer data for the other ones. But if that's what that means, then that might be a neat way of including the data on the other. Yeah.
Yeah.
Okay.
So because for Madurella mycetomatis, these compounds were much more potent than for falciformis spora senegalensis. We see that not only for this type of molecules, but we see that for almost every molecule. So there's something different about, well, of course, they are quite distant genetically, so that probably explains it. But unfortunately, there's simply so little known about falciformis spora senegalensis that we don't know that is how the cell wall would look like. We also don't know that for Madurella mycetomatis, but I guess there are major differences there, which simply prevents some molecules to enter the cell, which could enter the Madurella mycetomatis cell, simply because they are biologically different. Let's think of human and fish. That's the difference between the two. So it's really big difference.
Right. Okay. Okay. Yeah,
so
I don't
could well be that if some of these were tested in vivo against Madurella mycetomatis, they would have been active. But at some point, we simply needed to make a selection because we can't test all.
Yeah, sure, absolutely. No, yeah, absolutely. And that's why I wanted to include the actual numbers in gray, because it shows that lots of the other compounds are very potent.
Yeah.
The outlier I was talking about was this guy, which is one of the 4 that was taken on, and then the analog, this guy, which is the same apart from the chlorobromo, and it's just the difference is very striking between those. There's not. So the possibility is that something went wrong with the assay, or the possibility is that either molecule 526 or 527 aren't quite what we think they are. That's all. Unless we retest those, we might want to make a comment in the paper about it, because it's just really bizarre.
Yeah, so we test everything in triplicate in independent assays, so not on the same day.
Right.
So it could also be that something during shipment happened to that molecule.
Yeah, possibly, yeah. If you're seeing consistent biological data, then it's possible it's the molecule that's wrong.
I can take it with I'm testing this week some compounds, so I can take it with me if I find it in the freezer.
Yeah, you could take it, but it has been tested in triplicate, but we could take it along one more time to see if things changes, but...
Yeah. And in parallel, whoever made it could go back maybe and look at the NMR spectra. I can mention that too. To check the compound is what we think it is. Okay, great. Unshare. I'll tweak that diagram in that case. Just to reflect the traffic light thing.
Okay.
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 So
Sorry, go on,
ahead.
Wendy.
Oh, go ahead, Clementine.
I was just going to say, I have plenty of things to say about aminothiazoles. Some of it, I think, is, I don't need to discuss it with everyone if Matt has the time to talk about it. But what do I have? I have some compounds that I'll be sending tomorrow. We've had the long weekend here, so, slowed me down. But I'll send them tomorrow. I assume they'll get to you guys within like a week or so, which are just more aminothiazoles that have various substituents. And then the Kemble molecule, I have not a very catchy name, Kemble 472452, which Matt has flagged in the past as a known inhibitor of the same calcium channel that our lead compound in series 2 is a potent inhibitor of. So it'll be interesting to see if that works against mycetoma. And yeah, apart from that, I've been looking at writing up the aminothiazoles as a paper. So I have a bunch of specific questions about that. The main one is, previously for the phenaramols paper, you did like a big log D analysis, I guess. Why did you do that? And do you think it's worth doing for these compounds, essentially?
So that's interesting. So that was something that was noticed way back in the day by our then DNDi collaborator, Ben Perry, who I think because this must be something that he does that he did when he worked for DNDi, other projects, where he plotted the activity against log D. And noticed there was a correlation, it seemed, in the activity. And this was something that was for the in vivo active compounds. So the log D thing is the in vivo active compounds. That's the sort of that's where we're trying to apply it, really. And the rationalization that came as a result of that was this is something to do with compounds needing to get into the grain. And there's this apparent sweet spot of about 2.5, where it seems as though things get into the grain better if you're in that kind of region. What's the form of words that we put into the actual?
Yeah, maybe because we did that for this type of molecules, but it might be good to verify for another class of molecules to see if that
really good.
That would be the same, because I can imagine that it's also dependent on the type of molecule you put in.
Yeah, absolutely. So it seemed as though for the in vitro assay, log D values greater than 2.5 or better. But for the in vivo assay, log D values lower than 2.5 were better. So you kind of two competing factors.
Yeah, the idea behind that is that the fungus in vitro behaves quite differently than in vivo. So in vitro, of course, we expose fungal hyphae. There's no cement material, so there's no material produced by the fungus or the host which the molecule needs to penetrate. And also the metabolic state of the hyphae in RPMI media are probably different than the ones in the host. You see, if you look under the microscope, you see a very thick cell wall in the hyphae in the grain, which is absent usually in RPMI, where the fungus is happily growing. So it could well be that in vitro you do see a different characteristic of a molecule than in vivo. And that's also why we are aiming to develop this in vitro grain model, because there's no use in finding molecules which work very well in vitro but don't do anything in vivo. So we want to improve our in vitro models so that we can make a better selection.
But yeah, another series would be great, because I mean, presumably the in vitro tendency is in part related to how well the molecule is interacting with the target, the biochemical target, whereas the in vivo is governed to some extent by the grain itself. I mean, I'm assuming that's the case, but I don't know. But if you then did a different series with a different target, different biochemical target, you might see a different trend.
Okay, so Hung told me that they were basically statistically calculated then. Would that be something that we ask DNDi to do again, or
well, the
is
log
it...
D was calculated by DNDi, so the log D
Yeah.
values were. The statistical correlation is Wendy's day job,
Yeah.
right?
Yeah, I had a look.
That's not difficult for me to do. It's just copy paste. As long as I have a table, I can do that in GraphMap, not a problem. And for the log D, it was also not that difficult. I usually send an Excel file with the smiles to Ben. So now you could probably send that to Louisa. She puts that complete Excel file in her program, and the log Ds and all the other things you would like to know are simply added to the Excel sheet by that. The idea was that then at least we all use the same similar program because there are different programs, and then it would all be calculated in the same way. That was the idea behind it.
Yeah, that makes sense. Because I know you can definitely calculate log Ds with ChemDraw and other things, but I can put together an Excel easily to send to Louisa to get that on the way.
Yeah, great. I mean, yeah, log P is fine in ChemDraw, but log D it struggles with, I think. And that's why we asked Ben to, he had StarDrop, which was this software that he used, and which you have to pay for, and we didn't have access to it at the time. There are other bits of software that will do it too, which we, again, back in the day, we didn't have access to. But if Louisa has access to it, that would be the most consistent way of doing it.
Yeah, of course. Okay. I was also wondering, I might have missed this, but when we sent things for ADME testing a while ago, did we ever talk through those results? That was a while ago.
Yeah.
We sent a handful of compounds from each series. I feel like maybe Dimitri had it.
I think that was postponed all the time because then, yeah, first they were not there, then Matt couldn't join or something like that. I don't think they were ever discussed in the MycetOS meeting.
Oh, yes, that's fallen through the cracks, isn't it? Yes, we did get all of that stuff back, and I can't remember how many compounds from the phenomenal series we had done, actually. We certainly had others done, but I will dig up that spreadsheet and find out, because yes, we should, if we have the numbers, we should include them. Yeah, good catch.
Okay. I don't know if there's anything else that I am missing. I'm hoping, well, the compounds that I'm going to send tomorrow, I think I'll be able to write a solid discussion once I have some testing results from them. But at the moment, it's just going to be a fairly simple, we tested a bunch of compounds and we have a few basic, conclusions to draw, but there's not a whole lot of, I don't know what the word is, other angles to include in there. I'm assuming that I'm not forgetting something obvious that would be relevant. In terms of timeline, I'm assuming you guys are starting the in vivo testing at the moment, or like getting ready to start the in vivo testing in Erasmus? Okay.
Yes, so the culture is already in. The larvae are ordered, but next week we will start the first experiment simply
to train,
but also to see if the larvae and the fungus behave as they should be.
Okay, cool. So then hopefully we'll have some data for the compounds that I tested and then we're not 100% sure on those results. That'll, we'll get around to that fairly soon then.
Can you send me those compounds numbers so I can
Yes,
see
of
how
course.
much needs to be tested?
I think I ended up testing, I don't know, 8 or something, but I'm not sure that we would have done that because I started testing in vivo before we'd completely finished the in vitro testing. So possibly you should have a look at how many I tested and see whether you would have done them typically. Let's see, what else? Oh yes, I might just share my screen for a moment. So I was talking to a postdoc in the building who works with Michael Cassio, and they've been making these basically very similar molecules, aminothiazoles, for a completely different drug discovery project. I need to get Alice to talk to Michael, like as in the supervisors need to talk about whether they're okay with sharing this data, etc. But they have all of these compounds that they are happy, like the people who've made them are happy for us to test them. They just need to check with the boss kind of thing. I am not sure how much we think it is worth testing these because this would be tons of variations. I certainly don't think we'd want to do all of them. Mostly these ones don't have the pyridine, like they're not pyridines on this side, which hasn't suited our SAR previously. But the ones which are interesting, I mean, there's a few which are interesting, but specifically these ones, the 3 pyridines where the N is in different places. Previously, we tested compounds that had these N's in different places around the ring, but the left-hand side was not the same as our lead compound. So they weren't active, but we don't know exactly whether whether it was moving the N or changing the left-hand side. So if we can get those sorted, it'd be good to get those tested. We do, yeah.
Yeah, so Clementine, this number of compounds for in vitro screening only, it's not a problem. The high-low screening it fits actually in a 96 well plate. So if you select 3 or you select all of them for in vitro, that's not a problem.
It's
It
mostly
becomes
just that, yeah, I think a lot of these, I went through this previously. There's maybe like 6 or 7 variations on the right-hand side that I think would be worth testing, but then I don't know exactly the combinations that they have with the left-hand side. So it's, let's say 7 times, possibly, what's that? 11, but possibly fewer than 11.
Yeah, but we could also say we can triage simply by screening high, low, super low.
Super low.
So we are now also including often 2 micromolar so that we don't have to do the IC50 for too many compounds because that's more work.
Yeah.
And also for in vivo, that's the majority of the work. But you suspect, I hear from your message that maybe some of them will not be active and others will be. So if there's only a few active, then it would still be good to verify that the other ones are not active. Because that's also an answer.
Yeah, that's fair. Let's see. Basically, because these are what's the word? Mix and match. One of the green ones with one of the black ones. Can you see the thing that I've just opened? I don't know which screen share I'm using.
No.
The picture.
We see
Okay.
a red.
Let me.
Yeah, we see current library. The left side are red molecules, and now we see black molecules.
Yeah, sorry. I thought I had this somewhere, but I couldn't find it before. But these are the ones. These are the right-hand sides that I think would be interesting. So I guess there's actually 12 of them, and yeah, so that would be around 60 compounds that I think are worth testing. The other ones, there's no reason to expect that they would have any activity. So even though we can test some of them to see, it just seems a bit of a waste to measure them all out and
Yeah, for us
everything.
it's fine to measure these if you want to.
Okay, well, I'll see if I can get Alice and Michael to talk soon, because Alice has been away for a few weeks, so she's got a lot to catch up on. But yeah, hopefully I'll be able to get some of those to you guys soon as well. And let's see. What else?
The mechanism
Yeah.
of action. I mean, yeah, depending on what Michael is happy to reveal about the reason why he's made those compounds, it could be relevant to a potential mechanism of action. And I guess in the paper, we will still need to say something about that. So, what is known of the mechanism of action on any target of the most similar compounds. We're just going to have to summarize that. So if you put that into Campbell and stuff like that, then what is flagged up as aminothiazole potential target?
Yeah, I mean, I do have, to be honest, I'm not sure how much I am allowed to share versus not, in that,
Yep.
They just sent me a PowerPoint, but they do have some, solid information as far as I can tell, on those targets.
Right. Could be useful.
Yeah, definitely. I'll make sure that we ask about that as well then, instead of just sharing. Anyway, apart from that, I have a few very specific chemistry questions. Matt, I don't know if you saw, I was asking if we have a better way of naming our compounds, because 2-aminothiazoles is actually quite a unhelpful term to describe them. But I think that's everything on aminothiazoles otherwise.
Great. It's great you're driving this. It's great that something's happening with that series. We have Whit Gift School coming to UCL, I think, next week for their latest visit with a couple of compounds I think they've made.
Okay, wonderful. Yeah, we actually we have a high school student making one of them at Barker College, who I should actually probably email to see how they're going, because they'd done the first step when I last heard from them. And then I don't know if you saw, like, Anthony suggested that we try the methylated version of our lead compound.
Right.
And I don't know if you remember, Matt, the investigative reports that third year students have to do at UCID. But I convinced a student to do one of those, like to do that for his investigative. I don't know that he's done a great job of it, but if I can purify it, then hopefully we'll have that as well.
Okay, great. Okay. Sounds good.
I also have a chemistry question. I found a paper about the YOC1 target, and they found a compound that inhibited it in Candida. And I looked at the compound and I was like, oh, this looks very similar to some of the compounds we've tested. So I don't know who I should ask if they could make it, because buying it would be very expensive.
Oh, really? That's expensive, isn't it?
It's
Yeah, that's
5
the
milligrams for more than 300 euros.
And you've posted that one in this meeting, haven't you?
Meeting. Yeah, I was hoping that Peter would join because he goes for series.
Right.
And I mean, Matt, you would know better than me, but the phenothiazines aren't particularly complicated to make, are they?
No, they're fine. So, I mean, maybe if you tag Peter and tag him, he might then see it and respond about whether we could access that molecule. Is it just that one, or is that part of a series?
Yeah, there was also another one, but they didn't really have good evidence that it inhibited it. It was an acetyl, similar to that, but an acetyl.
Right. Okay. I mean, if they are expensive, so a molecule that is like that is good value for money would typically be like 100 euros for a few milligrams. Did you say it was like 300?
More than 300 for 5 milligrams.
Yeah, I mean that's sort of only about roughly twice the price of if you buy a molecule from the enamine. So it's not actually that expensive for something which, but still, I know it's still expensive. It's just not that expensive. But if you can't, if it's not, available at a good enough price, then, and if we can't convince Peter to make it, then The other thing would be to see if anyone in the last, 3 years or something has made a molecule like that in like a medicinal chemistry paper. And then you could reach out to them and say, hey, we're looking for a sample of this. Do you happen to have any in the fridge, kind of thing?
Okay.
More than a few years, it's very unlikely to work. But if it's something recent, then you might get lucky.
Okay.
That would mean finding the molecule in something like SciFinder. I don't know if you have that, or PubChem or something like that, to try and find someone who's made it recently. If you don't have a resource to do that, then I'm sure that Clementine and I could do that, because we have access to things like SciFinder, which tells you exactly who's made which molecule and when.
Yeah, I mean, it doesn't, well, if it was really interesting, I'm sure that we could get it made here at UCID, but certainly it might take us a while. I have sorry, I only know the name that I've been referring to it as, that molecule which had 3 pyridines on it, Dunya, that you found in another paper.
Yeah.
That little triangle one. I did actually make that, but I haven't been able to purify it. So I didn't want to say, I've made it, because I think I will remake it and try and purify it again.
Okay.
And send it to you in a later batch.
So the last point I put on the agenda was since last month, not last month, the month before that, we discussed about the targets, the new targets. Are there any thoughts on that? Matt, are you already aiming to start somebody to make something, or?
Yeah, so I put that into a proposal to this fail-safe grant scheme, saying that we needed some resource to make the proteins and then run screens on those in the SGC. And so we didn't get that, but the feedback from them was that we were too early stage for their main grant scheme. So we needed to be more advanced, basically. But that I could submit something called for a seed corn award, which is much smaller, but which would allow us to make protein. And I didn't manage to get that in by yesterday, because I've been on vacation the whole time, and it was just absolutely impossible. And they also require for that a fairly significant industrial contribution. So something that comes from industry and a person to be named from the company, and for them to provide a CV and everything like that. And that's actually quite hard in the fungal area to get someone who can do that and provide something. So got some ideas about what we could do there, but I didn't get it in. The thing we need here, I think, is to act on our shortlist of targets and try and get some of those proteins made. And I just don't know how best to do that. Because if we got the protein made, we could then run a screen to see if we can find binders. I think the list is quite good that we got. I mean, I think that's actually quite good as a starting point. But maybe for a grant proposal, we need to actually do a little bit of the preliminary work in advance.
Yes, no, I did make proteins in the past. I did that in E. coli. So we certainly have the knowledge and the things we need to do that.
Right.
But it would be good just to agree, all of us, on which molecule to go for, or which protein, because for us to purify all of them, it's still quite a lot of work to do. But if we just simply go for 1 or 2, that's relatively easy to do.
Yeah. So it seemed like, I mean, the 2 kinases in Mickey's paper and the YAC-A homolog that we were talking about, those seemed attractive, right?
Yes.
Because they'll probably express, I assume they express okay, because they're not completely out there. I mean, they have homologs which may have been expressed already, kind of thing. It seemed like that would be a good one to go for, because they are also relevant to the development of the grain. So it's not just finding another target, it's trying to find a target that would in some way address the problem of well, not resistance, but the problem of the barrier to developing effective medicines. So it seemed like that was a strategically good one to go for.
Yeah, and it seems like all the things we're currently doing in our lab, so these are not the only manuscripts we're constructing. Ma did screen 5,800 molecules, and again kinases popped up, kinase inhibitors. So it seems like the kinases really are very interesting to look a little bit further into. And these, at least those two from Mickey's manuscript, are in the same pathway. So that would also be something we could target later on with knockouts when Tim has get the methodology working.
Right.
So yeah, these were the things I was thinking of. Maybe let's go for those kinases, because at least they are enzymes, so it's more likely to be inhibited.
Yeah, yeah. So I mean, I don't know how much work that is, making those, but like I said, for the screen that we would want to do, we would just need about 2 milligrams or something of it, with this his tag on it, so that we can immobilize it. So I don't know how well they express in E. coli. I don't know what kind of yield you get and how difficult it would be. But if that could be done,
the TCTP molecule I did, the TCTP protein and the fructose bisphosphate protein, they were quite easily to purify and actually get in high amounts. So we don't have to take off the histidine tag then. That's easier.
Yeah, right. We need that for right, immobilization. Yeah.
Yeah, so what we normally do, we design the plasmid with an histidine tag, and express it in E. coli and use a nickel column to purify.
Right. Right. Yeah. So the same tag is the thing that's used to attach it to the bead for the screening. So works pretty
so
well.
what I could do is I could write a intern proposal for it. So then the intern could work on that in 4 or 5 months from now. And if we can do it earlier, maybe we can, but I don't know, because the larvae season is starting, so I don't want to put more things on Dunja.
The next PhD student can come in swinging.
Yeah, but that is a different topic. Yeah. So that's also, but then we could make it an intern project, because I don't think you can do it earlier with your partners either. Is that correct?
Yeah, I mean, it would take a while. If I mean, I have to triple check that when the protein is made, that we can then run a screen on it, that there is capacity for doing that. There's only a certain amount of capacity, and you've got to make sure that we're not assuming that there's too much. So I'd want to do that. So if we know it's coming, then I can make sure that there's capacity to run it.
Yeah, but that probably, because if it's an intern project, then probably it will not be before the end of this year that the protein is there. But yeah, we could always give it a go, and if it's not this year that it can't be screened, then it might be the next year.
Yeah.
But as long as it's in E. coli and we know that it expresses, then it's not that difficult to express it at any time we want to.
Yeah. Right. Okay. I mean, in the meantime, we should keep our eyes open for other opportunities for resource to make proteins, because I think that is the key bit here. We've got to try and show that we've got starting points against new targets, and I think that is going to be crucial. It's either that or it's again phenotypic screening, and I think that you've done a huge amount of work on the phenotypic screening side, which has yielded great things. But I think there is a lot to be said for going after novelty on a more targeted approach than that.
Yeah, and I think that should also be the next thing, because now we screened, if we add everything up, roughly 10,000 compounds.
Amazing.
So if we screen another 10,000 or another 100,000, I don't think we will get that many new clues.
Right.
And of course, we can remine the transcriptomic data we have to find new pathways, but maybe it's good to have at least a proof of concept.
Yeah, exactly. That's how I see it. And a proof of concept where we're targeting the Madurella protein, but you could also imagine taking what we find and developing those molecules against other fungal pathogens, right? So things that aren't neglected, because I think that's the way to get industry interested, is to go after something new like that and say, well, of course, this could be taken in one way for mycetoma and in another way for something else.
Yeah, and those two kinases in Mickey's paper, they are also in Aspergillus and Candida, so
Right.
those can be repurposed for that.
Yeah, that would be very good.
Yeah. Okay.
I'll tidy these minutes up later. But have we got any other subjects to discuss, or?
I think that's everything. I mean, it's the three papers, isn't it? And it's the aminothiazole stuff, and it's the proteins as new target stuff. I mean, I think that's everything.
Yeah, and you might also be glad to hear, Matt, that somebody is making those OSM compounds now. So,
Oh yeah.
I mean, he just started, but hopefully they'll be sorted soon.
Oh, cool. Okay. Yeah, that's all I
Alrighty.
had.
Yeah, and before we go, with our meeting next month, who is writing the agenda for that?
When is next month? Just double checking, because
For
Dunya and
May.
I might be in
in Brazil, yeah.
Ah,
Yeah, the conference of the International Society is actually of Human and Animal Mycosis is in Brazil this year. It's only once every 3 years, but
It's the week after Brazil.
it's the week after Brazil, so we can give an update on Brazil then too.
nice.
You make us jealous. with
Yeah.
Well, Dunya will have a poster on MycetOS.
Yes.
There will be a session on MycetOS too. D&DI will be there too. So,
Okay.
Very nice.
yeah, I can take things there. If we need something, please let me know. I can discuss with Borna at least.
Right.
Yeah, that's
Yeah, I mean,
great.
so that's where Louisa is based, right?
Yeah,
I
but I think it's Borna actually, the head of the Mycetoma program in DNDI, who's going.
Oh,
But
wow.
maybe Louisa will pop by. I don't know.
Pop in Brazil.
Yeah, it's not in one of the main cities. It's in Iguazu Falls.
Oh, wow. Lovely.
Yeah. So we decided to take on one extra day to see the falls, actually.
Amazing.
When in Rome.
Yeah.
Yes.
Yeah.
I happen to know about that because we went there. And one side is definitely better than the other. Pretty sure the Brazilian side is better than the Argentinian side, or whatever it is.
Well, that's
Yeah.
good news.
Yeah, I think that's right. Because in one side you can actually walk around them, and in the other you're looking at it from a distance.
Yeah, I think
so
we're the part where you look at a distance. But
oh, okay. I'll try and remind myself which one we saw, because it was a strange trip. We were there for a couple of days. Definitely worth seeing though. It's amazing.
Yeah, I've never been there, so yeah, curious to see.
Wow, great. Okay, sounds good.
Okay. Well, we'll see you when you're freshly back from Brazil, I guess.
Okay.
Okay, good luck with the rebuttal.
Yeah, thank you. And you with the submission.
Yes. Okay, take care, everybody.
All right. Bye-bye.
Bye.
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 No.